Hello, everybody. I'm Kumar Shalupai. I'm CEO and CSO of Tiziana Life Sciences. Tiziana Life Sciences is a clinical stage biotechnology company. It is listed on NASDAQ. T symbol is TLSA, and it is also listed in London Stock Exchange, and symbol is TILS. I'm delighted to be with you today to give you some very important updates on our clinical program, as well as some business development areas. So let me take you to slide number 3. And the purpose is just simply to give you updates. I'm not going to go through the whole presentation, but just give you some important updates on some of the recent clinical updates, as well as some business development programs. So as it is listed on the slide, the most important update is that this morning we executed an agreement with Precision Bio. Precision Biosciences is a biotechnology company engaged in CAR-T applications for cancer treatment. CAR-T is, as you may already know, is very important innovative technologies for cancer treatment. And this CAR-T has potential to revolutionize the way cancer treatment is conducted. So we signed an agreement with Precision Bio for them to use our Foralumab. Foralumab is a fully human anti-CD3 antibody. And this antibody is unique in the sense that it is well positioned for use in CAR-T application and potentially to improve clinical outcome as well as long-term success. I'll go in a little more detail a little later in my presentation. In addition to this agreement, we also completed and publicly reported data on use of nasally administered Foralumab for treatment of COVID-19 patients. The detailed results were published, and we announced that publicly last week. And this was the first ever validation of nasal administration of Foralumab for treatment of COVID-19. And as you will find a little later, that this is a unique approach which may have broad utility for treatment of all variants of COVID-19. We also completed 2 phase 1 trials with oral and nasal administration, and phase 2 studies with oral and nasal are going to start very soon. Phase 2 study with Milciclib in sorafenib-resistant hepatocellular carcinoma was completed, and we reported data publicly. The results are fantastic, and now we are looking forward to starting the next step. While we were doing this, we also found out that Milciclib may have potential for treatment of KRAS positive NSCLC, which is a huge opportunity for us, and we would be announcing that, initiating that trial pretty soon. Let me take you to slide number 4. What I want to emphasize here is that Tiziana Life Sciences is trying to switch in intravenous therapy to alternative routes of therapy, such as oral administration, nasal administration, and inhalation administration. The common theme in this alternative route of antibody administration is to be able to facilitate local or topical action. And because you're providing antibodies such that it may have local action, the amount of antibody needed to provide clinical benefit may be very low. And therefore, these alternative routes could maximize efficacy while minimizing the toxicities. Other important thing about these alternative routes of administration is that intravenous treatment with antibodies or immunotherapy has been very successful. However, the compliance is poor because patients have to go to doctor's office every 2 to 3 weeks to refuse 2 to 3 hours of infusion. And more importantly, it is also a very costly affair. As far as toxicities are concerned, it has been seen that when antibodies are given through intravenous administration, the drug level in blood is very high for the first couple of hours, and that leads to the toxicities. So in By switching to oral or nasal or inhalation administration, we believe we may be able to overcome these toxicities. Let me take you to slide number 5. I want to focus or emphasize here that anti-CD3 antibodies has been in clinical for a number of years. And just to remind you that antibody shown in red color on the left is called OKT3. This antibody was developed by J&J and it was approved by FDA. But because it's a fully mouse anti-CD3 antibody, it produced severe immune reactions, and there were also anti-drug antibodies developed. Therefore, it could not be used for long term. So this antibody was taken off the market. But clinical efficacy was so good that number of companies tried to humanize this antibody, and I've given you some examples here, particularly teplizumab and visilizumab, shown very good clinical efficacy. Unfortunately, both of these antibodies also produce anti-drug antibodies. I'm very pleased to tell you the Foralumab is a fully human anti-CD3 antibody. So far, we have not seen any immune reaction, and we have not seen anti-drug antibody development as well. So therefore, Foralumab is well positioned for long-term use, and it seems to be very safe antibody. Therefore, it could have potential in a number of diseases as listed here. Our focus is on with nasal administration for multiple sclerosis, oral administration for Crohn disease, and also, as we announced this morning, the intravenous or subcute administration for CAR-T. We are also moving forward with other potential applications, but our current focus is for these diseases. Before I go in more details of other programs, let me also show you this slide number 6. This is the clinical data from the trial that we completed in Brazil. And this trial had 39 patients. And this trial was the first ever trial completed with nasal administration. And the purpose was, and in fact, this came out from our phase 1 trial with nasal administration of Foralumab, where we saw that treatment resulted in upregulation of T regulatory cells. T regulatory cells are body defense. And in COVID-19, we see that T regulatory cells or Tregs are depleted. So therefore, we thought that nasally administered Foralumab may be able to upregulate Tregs and thereby strengthen your body defense to fight with the COVID-19 disease. And I'm very happy to tell you that this once-a-day treatment with just 100 microgram per day for 10 consecutive days produce very strong results as shown here. This article is already published here, but what I want to emphasize here, that Foralumab alone reduced lung inflammation by 80%. I mean, in 80% of patients showed rapid improvement in lung and reduction in inflammation. And this was very well supported by the biomarkers of inflammation. We looked at IL-6 as well as CRP. Both are very important biomarkers for inflammation. And what you see here is that Foralumab treatment resulted in reduction of these biomarkers in 69% of patients and 85% of patients. This is very strong indication that, yes, Foralumab is working. And it was also observed that the sense of smell is lost when COVID-19 disease occurs. And this sense of smell was recovered within 3 to 4 days. So this is very, and first ever validation that antibodies can be given nasally. And we are very pleased with it. Now we are moving forward and hopefully we'll announce very soon, starting the next phase 2 trial in COVID-19 patients in Brazil. This slide is mainly to tell you where Foralumab could be used in CAR-T. So this slide shows you different processes leading up to development of CAR T cells. CAR T cells are engineered in such a way, or they are supercharged in such a way that they go and kill tumor cells, which is a unique approach, and potential of this technology is huge. One limiting factor, however, is that CAR T cells, because they are engineered or they are modified, so when they are injected in human body, our own immune system attacks these cells, and their survival is very poor. And as a result of that, the recurrence rate for cancer is very high. There have been many strategies to increase or enhance survival of CAR T cells, but so far not much success in that field. Foralumab is a fully human anti-CD3 antibody. Foralumab is known to cause lymphodepletion, which is what is needed for increased survival of CAR T. So this approach, I patented it, and that is where Precision Biosciences is interested in using Foralumab for enhancing their CAR T therapy for cancer treatment. In addition to Foralumab, we also patented the co-stimulation with other drugs such as anti-IL-6. Let me also tell you that we also have a very unique, fully human anti-IL-6. And there have been a number of very recent findings that anti-IL-6 antibody may also work along with lymphodepletion agent to enhance success, as well as for long-term success of CAR T. Therefore, we believe it can be used for CAR T as well. But our patent also covers use of inhibitors of PI3 kinase. It's a pathway that gets upregulated during these processes, and inhibition of that pathway could also be very good for CAR T. So we are very happy and delighted that there is now a novel avenue and a huge opportunity for Foralumab to be used for cancer treatment. Let me take you to slide number 9. This is the clinical data from Milciclib. This we reported publicly. And here, the clinical data suggests that Milciclib in sorafenib-resistant hepatocellular carcinoma patient showed fantastic results. And I can tell you with comfort that Milciclib monotherapy seems to be outperforming the existing therapies for hepatocellular carcinoma treatment. We are moving forward on this program as well. And while we were doing that, we also found out through animal studies that Milciclib may also have potential for treatment of KRAS positive NSCLC. KRAS positive NSCLC is in a way still unmet medical need, but about a couple of months ago, a drug from Amgen showed benefit to these in treatment of these patients, and that drug is already approved now. The market is so huge that we think that Milciclib may still have opportunity there. So that gives you the overall overview of our clinical programs. And the next slide, slide number 11, shows the progression of Foralumab. As I told you before, that nasally administered Foralumab is already being evaluated under the expanded program for multiple sclerosis, and the patient has already completed 3 months. And so far, there are no safety issues, and that's very good news for us. Oral capsule for Crohn disease, we should be announcing very soon, starting this trial. We completed intranasal phase 1 exploratory trial in COVID-19 patients, and now we are moving to phase 2. In addition to this, TLSA, which is anti-IL-6 antibody, most of the processes have been completed, and we are waiting for a few more studies to be completed, and we should be announcing submission of IND by the end of 2021. Thank you very much for listening and showing interest in Tiziana Life Sciences and for your continued support. If you need more information, please visit TLSAinfo.com.